He was carried into his tomb on a January morning nearly 300 years ago. A young man no older than 19, his body wrapped in linen, his face covered in gold, his legs broken and badly healed, his chest caved inward from something the embalmers could not fix. 130 walking canes lay beside him. Not ceremonial props, not symbolic offerings. Actual, worn, functional canes, the kind a person uses every single day because standing upright without them is agony. His left foot was clubbed, his spine was curved, his skull was slightly elongated, though not from ritual binding. A malaria parasite, Plasmodium falciparum, had been threading through his blood in the final weeks of his life. He was a teenager. He was the most powerful person on Earth, and he could barely walk. This is not the Tutankhamun the world inherited. For a century after Howard Carter cracked open the sealed door of KV-62 in November 1922, the story of Tutankhamun was written in gold, in the 398 objects that surrounded him, in the painted walls of his burial chamber, in the elaborate funerary mask that became the defining image of ancient Egypt. Every museum placard told the same version. Every documentary repeated the same arc. A boy king, likely murdered, possibly by a scheming advisor named A, possibly by the General Horemheb, possibly by a blow to the head from enemies within the court. The golden mask showed a face of divine serenity, perfectly symmetrical, strong, eternal. The established narrative was seductive in its clarity. Tutankhamun was a pawn, a symbol, a mystery solved by looking at who came after him and who might have wanted him gone. Physical archaeology supported this reading more or less. The tomb itself was smaller than those of other pharaohs, hastily finished, the paint still damp in places, as though the decorators had been working against a deadline. That suggested an unexpected death. A skull fragment found inside the burial cavity suggested a possible blow to the head. For decades, the murder theory was the dominant frame. It was a tidy explanation. Logical, supported by tomb architecture, funerary evidence, and the suspicious political speed with which I assumed the throne. Scholars agreed. Textbooks said so. And the mystery of who killed Egypt's boy king anchored countless documentaries, books, and museum exhibits across the 20th century. Then, a genetics laboratory quietly dismantled the whole thing. In 2010, a team led by Zahi Hawass, then Secretary General of Egypt's Supreme Council of Antiquities, alongside molecular geneticist Karsten Pusch of the University of Tübingen, published a landmark study in the Journal of the American Medical Association. The paper, titled Ancestry and Pathology in King Tutankhamen's Family, was based on 4 years of work, involving 11 royal mummies from the New Kingdom period, including Tutankhamun himself. The challenge was staggering. Ancient DNA is not like the clean double helix spirals drawn in biology textbooks. It degrades, it fragments. In a hot, dry climate like Egypt's, where temperatures inside tomb chambers can swing dramatically, genetic material breaks down over millennia into microscopic shards, like trying to reconstruct a shredded novel from confetti scattered across a football field. The team worked in a purpose-built, sterile laboratory constructed inside the Egyptian Museum in Cairo, using a protocol so careful that each mummy was sampled in a sealed environment before any other researchers or visitors had access. Cross-contamination was the enemy. One stray modern skin cell could corrupt an entire ancient genome sequence. What they extracted from bone, tooth root, and soft tissue was mitochondrial DNA and autosomal markers. Mitochondrial being the genetic material passed only through the maternal line, like a cellular heirloom passed mother to daughter across thousands of years, and autosomal being the full chromosomal picture inherited from both parents. They ran the samples through short tandem repeat analysis, a technique used in forensic identification that examines specific locations along the genome where sequences repeat at variable lengths. The sample sizes were small, the fragments were damaged, but the signal was undeniable. What they found didn't match the archaeological narrative, not even close. Based on the historical record, geneticists had expected to find evidence of a carefully maintained royal lineage. Powerful, yes, possibly inbred to some degree, as Egyptian royal families were known to practice marriage within the dynasty, but essentially a coherent genetic picture reflecting deliberate dynastic continuity. Instead, they found something that made the physical archaeology suddenly look like a symptom rather than a story. Tutankhamun's parents were full siblings, not half-siblings, not cousins elevated to the status of brother and sister in ceremonial language, as Egyptologists had long assumed when royal inscriptions used familial terms. Full biological siblings, father and daughter, or brother and sister. The genetic data could not distinguish between the two configurations, but it was unambiguous on the central point. His parents shared both parents in common. Here's where it gets strange. The likely father was Akhenaten, the heretic pharaoh, the revolutionary who dismantled Egypt's pantheon and replaced it with monotheistic sun worship, who built a new capital city from scratch in the desert and forced the entire Egyptian court to relocate to it. Akhenaten's identity as Tutankhamun's father had been suspected archaeologically. The DNA confirmed it, but Tutankhamun's mother, the unnamed woman whose mummy, designated KV35YL in the archaeological record, was found in a side chamber of another pharaoh's tomb, was not Nefertiti, not the famous queen whose bust sits in the Neues Museum in Berlin, whose profile is the most reproduced face in ancient Egyptian art. The genetics showed that Tutankhamun's mother was a full sibling of Akhenaten, a sister who had also become a wife. No name, no surviving inscription that clearly identifies her, no ceremonial record. She vanished from the historical accounts so thoroughly that researchers now believe her existence may have been deliberately erased. Her name chiseled from walls, her image struck from monuments, her identity buried as a consequence of the very union that produced Egypt's most famous king. But she is there, in the DNA. Impossible to erase from cells, the consequences of that union were written into Tutankhamun's body with brutal precision. The study identified a minimum of 2 distinct pathological conditions, consistent with the inbreeding coefficient, a measure geneticists use to calculate the probability of inheriting identical gene copies from both parents, like rolling a weighted die weighted toward genetic repetition. Tutankhamun's coefficient was exceptionally high. Not a trace of the usual buffering that genetic diversity provides, not background noise from occasional cousin marriages, a substantial founding collapse of heterozygosity, the biological term for having different gene variants at the same location, which is what protects organisms from recessive genetic conditions. When that diversity disappears, recessive traits that would normally be suppressed by a dominant healthy variant get expressed. Both copies say the same broken thing, and the body listens. Kohler disease, a rare vascular bone necrosis, the death of bone tissue due to interrupted blood supply, was identified in 2 of the bones in Tutankhamun's left foot. This, combined with a clubfoot deformity, explains the 130 canes. He was not merely hobbled. Walking was painful in a way that left physical evidence on the skeleton itself, erosion patterns on joint surfaces that told researchers his gait was fundamentally compromised. Additionally, the team identified Freiberg-Kohler syndrome in the right foot, another form of bone, death affecting the metatarsal, multiple overlapping structural conditions. In both feet simultaneously, not one fracture, not one bad fall. A systematic failure of a skeletal integrity expressed across the lower body. The exact biological outcome you would predict from extreme inbreeding playing out in real anatomy. And then there was the malaria. Plasmodium falciparum DNA was detected in 4 of the 11 mummies studied, including Tutankhamun. The team found evidence of multiple infections in his body, meaning he had survived earlier malarial episodes before what may have been a final fatal bout. Malaria in ancient Egypt was not unusual in the broader population, but combined with a severely weakened immune system, compromised bone structure, and limited mobility, it becomes something different. It becomes a body that had very little left to give. The murder theory evaporated, not weakened, not complicated, dismantled. A skull fracture, it turned out, had occurred post-mortem, during the mummification process itself, or possibly during Carter's own excavation in 1922. The blow to the head that mystery writers and documentary filmmakers had built careers around was an artifact of rough handling, not a royal assassination. Eye and Horemheb had not killed Tutankhamun. Biology had. Slowly, inevitably, from the moment of his conception. But that's not the remarkable part. The remarkable part is what the DNA revealed when the team ran genetic comparisons between Tutankhamun's profile and modern population databases. Haplogroup R1b1a2, a Y chromosome lineage, was identified in Tutankhamun's paternal line. This haplogroup is rare in modern Egypt, where it appears in roughly 5 to 10% of the male population. But in Western Europe, particularly in populations from the British Isles, France, Spain, and Germany, R1b1a2 is the dominant Y chromosome lineage, present in 50 to 70% of men. The data was striking enough that a Swiss genealogy company, iGenea, independently analyzed a partial genetic profile derived from footage of Hawas's team at work and estimated that approximately 70% of British men and half of all Western European males share a common paternal ancestor with Tutankhamun, tracing back to the Black Sea region of the Caucasus, somewhere between 9,500 and 7,300 years ago. The findings were not without controversy. Hawass disputed the iGenea analysis, arguing it was derived from non-peer-reviewed documentary footage rather than raw sequence data. The broader scientific community was divided on the robustness of specific haplogroup identifications from such degraded ancient samples. And yet the core finding, that R1b1a2 or a closely related lineage was present in the royal line, has not been disproven. What it suggests, cautiously, is a population connection between the ancient Near Eastern and European populations that predates pharaonic Egypt itself, a lineage threading through the Caucasus and into North Africa via migration pathways that geneticists are still mapping with each new ancient genome project. Comparing 299 ancient genomes from across the ancient Near East, North Africa, and early European populations, as researchers like Johannes Krause at the Max Planck Institute for Evolutionary Anthropology have done in related work, reveals a picture of the ancient Mediterranean world defined not by fixed, isolated populations, but by relentless movement, convergence events, admixture pulses, populations meeting and merging across the span of 50,000 years of history recovered from dental pulp samples no larger than a grain of table salt. Tutankhamun's genome does not sit outside that picture. It sits inside it. He was not simply an Egyptian pharaoh in the nationalist sense that modern categories project backward onto ancient demographics. He was a biological product of movements and mixtures that preceded the dynasty, preceded the language, preceded the religion he had briefly dismantled and his court quickly restored after his death. Every strand of his DNA connects outward to populations across North Africa, the Levant, the Caucasus, the European steppe. We carry pieces of these people in our cells, not metaphorically, literally. Written into the Y chromosome of roughly half the men in Western Europe is a lineage that branches from the same ancient trunk as Tutankhamun's paternal line. Written into the autosomal DNA of millions of people across the Mediterranean, the Middle East, and North Africa are fragments of the migrations and admixture events that shape the new kingdom's genetic landscape. When modern individuals send saliva samples to ancestry companies and receive results showing traces of North African or Caucasian ancestry from deep prehistory, they are in some small molecular sense touching the same ancient human currents that flowed through KV62, and the implications reach further still. The pattern of extreme royal inbreeding identified in the 18th dynasty is not unique to Egypt. Comparable levels of inbreeding with comparable physical consequences have been identified in later dynasties, in certain periods of European royalty, and in isolated island populations. The biology does not care about the political rationale for dynastic purity. Recessive conditions emerge with mathematical reliability. Infant mortality rises. Physical robustness declines. The same genetic story plays out across cultures, across continents, across centuries, a recurring demonstration that no political institution can permanently insulate itself from biological reality. Albert Zink, director of the Institute for Mummy Studies at Urac Research in Bolzano, Italy, who conducted subsequent CT scanning and isotopic analysis of the royal mummies as part of ongoing investigations, has described the Tutankhamun case as a template for understanding how ancient power structures wrote themselves into bodies. The physical condition of the king, Zink noted, reflects not just individual circumstance, but systemic dynastic practice. What the genetics revealed was not merely one young man's suffering, it was the biological ledger of a political system, a record of what happens when the drive for dynastic continuity overrides the genetic diversity that living organisms require to thrive. This isn't just about one pharaoh, this is about the price of power maintained through purity, a human pattern that repeats across the archaeological record with devastating consistency. Somewhere in the basement of the Egyptian Museum in Cairo, behind climate-controlled glass, under carefully calibrated lighting, the golden mask still stares outward, serene, perfect, eternal. The face that launched 1,000 expeditions, 1,000 exhibit labels, 1,000 documentaries insisting on murder, on intrigue, on a boy king struck down by scheming courtiers. It holds answers to questions we are still learning to ask, but it also raises new ones. If the man behind the mask was this compromised, this physically reduced, this biologically isolated, how many other ancient rulers were carrying silent genetic burdens that their official portraits were designed to conceal? How many more royal mummies lie in museum collections and tomb chambers across Egypt and the ancient Near East, waiting for sequencing technology to read what their bones have always been saying? You can erase a name from stone, but not from cells. You can commission a mask of gold and demand that every image depict perfection, but the skeleton will tell the truth regardless. Tutankhamun's body was a library, sealed for 3,300 years, catalogued by Carter in 1922, and only now, in the 21st century, being properly read. Civilizations are more fragile than we imagine. Most human stories vanish completely. We see only the ones who left descendants, who left inscriptions, who left enough biological material to survive 3 millennia of heat and time in the rough hands of excavators. Tutankhamun survived because he was buried in gold, because his tomb escaped the systematic looting that emptied most of the Valley of the Kings, because his body was preserved with sufficient care, that a fragment of ancient DNA could survive inside a molar root and speak, 3,000 years later, to a geneticist holding a sterile extraction tool in a sealed laboratory in Cairo. The story of Tutankhamun is not finished. It is only beginning. As whole genome sequencing of ancient samples becomes faster and cheaper, as new mummies are discovered and existing collections are re-examined with modern tools, we will rewrite entire chapters of this history. The 18th dynasty is not the endpoint of this inquiry. It is the entry point. There are thousands of stories like this one, sitting in museum drawers and laboratory freezers waiting to be told. Subscribe so you don't miss what comes next.